Welcome to today's Explainer. We're opening up our researcher's notebook to dive right into the bleeding edge science of how a single amino acid acts as the ultimate cellular switch. Yep, we're talking about cysteine. In the world of signal transduction, we're so used to thinking about binary on and off switches, like phosphorylation. But cysteine, it doesn't play by those binary rules. It actually occupies this uniquely privileged position in cell signaling, possessing an incredible structural and chemical flexibility that, honestly, scientists are just now beginning to fully map out. So, as researchers, how do we decode this ultimate biological switch? Well, to figure that out, we first have to set up the biochemical mystery of cysteine itself. I mean, why this specific amino acid out of the 20 we have available? It all comes down to its unique sulfur atom and the reactive thiol group attached to it. Now, normally, the pKa of a free cysteine thiol is around 8.3. But when it's tucked inside the specific microenvironments of a protein, maybe surrounded by basic residues or hydrogen bonds, that pKa can drop dramatically. We're talking down to between 4 and 6. And since physiological pH hovers right around 7.4, this massive drop transforms cysteine into a hyperreactive thiolate anion. It essentially becomes this highly tuned biological antenna, uniquely positioned to sense and integrate incoming chemical signals from all across the cell. Just look at the sheer versatility of the cysteine modification spectrum. Cysteine can wear multiple chemical disguises, serving as this complex multi-state redox code. It's literally integrating reactive oxygen, nitrogen, and sulfur species simultaneously. It can be oxidized into sulfinic, or sulfonic acids. It can form disulfides, undergo S-nitrosylation, S-glutathionylation, or even persulfation. It's wild. A single residue acts as a dynamic, multi-input sensor, fundamentally altering the function of the protein it lives in, based on the exact environmental stress the cell happens to be experiencing at that moment. So the absolutely crucial point here is reversibility. Because think about it, without a biological reset button, these modifications would just equal permanent cellular damage. But biology is way more elegant than that. Cysteine modifications are specifically designed to act as dynamic relays, and they absolutely require this exact reset mechanism to function. We actually have dedicated structured relay machineries, specifically the thyredoxin, glutaredoxin, and peroxyredoxin systems, and they work around the clock to reverse these modifications. They ensure that the moment a signal is sent, the switch gets flipped right back to its resting state, which gives us precise temporal control over the whole signal relay. And this brilliantly illustrates that exact reset button in practice. Take a look at S-glutathionylation and its GRX1/2 reversal mechanism. When a cell is under heavy oxidative stress, its proteins are at serious risk of irreversible damage. But by forming a mixed disulfide with glutathione, the sensitive cysteine is temporarily shielded. It's protected. Then, once the stress passes, enzymes like GRX1 and GRX2 sweep right in to specifically reverse that mixed disulfide, putting the protein back to its original state. It's just an incredibly precise protective mechanism that our cells completely rely on to survive in constantly changing redox environments. Okay, let's dive into this head-to-head comparison, because the exact nature of these modifications drastically changes the cellular outcomes. On one hand, we have sulfonic acid. This is an unstable intermediate state driven by reactive oxygen species, like hydrogen peroxide. It acts as an electrophilic acceptor and honestly often serves as a pivot point leading to other modifications. Now, contrast that with persulfidation. This is a robust, uniquely potent signaling modification driven by hydrogen sulfide, which is a gaseous signaling molecule. Persulfides are actually way better reducing agents and nucleophiles than standard diols. By looking at these two side by side, we can really see how the cell uses completely different chemical logic to route oxygen-based signals versus sulfur-based signals right through the exact same amino acid. Moving into our next section, Lanzara's paradigm shift on receptor biophysics, where we look at moving beyond passive redox sensors. So we've spent some time looking at the redox chemistry, which, is the dominant view in the field. But now let's pivot to a fascinating second axis of cysteine signaling. This is the revolutionary work contributed by Richard Jean Lanzara, which totally expands our view of cysteine far beyond simple passive biochemistry and pushes it straight into the active realm of GPCR biophysics. Lanzara's framework introduces a radical shift in how we view receptor activation. Let's walk through this theory step by step. Traditionally, we think of a ligand docking into a receptor like a key slipping into a lock, with the receptor just passively changing shape. But this work argues that cysteine isn't just sitting there reacting to ambient cellular stress. It's an active structural participant in the intrinsic chemistry of receptor activation itself. When an agonist binds to the GPCR, reactive or uniquely positioned cysteines near the binding site actively dictate exactly how that ligand docks, fundamentally shaping the receptor's structural transition right into its active state. To really see this in action, it helps to ground the theory in one of the most rigorously studied receptors in all of pharmacology, the beta-2 adrenergic receptor. Structural and mutagenesis studies consistently highlight a highly conserved cysteine residue at position C3.25. And what's so fascinating here is that this specific cysteine is repeatedly shown to be absolutely critical for receptor function. It essentially acts as a conformational gate, actively dictating agonist efficacy. It's a perfect textbook illustration of the Lanzara-linked framework. These local cysteine interactions are literally pulling the levers of the macroscopic pharmacological response. And this brings us to a really powerful concept, captured perfectly in this quote. Initial receptor response creates the net shift in molecular efficacy. This completely reframes how we think about receptor binding. Instead of a static lock-and-key model, we're now looking at this through the lens of Le Chatelier's principle. Ligand binding perturbs the receptor's conformational equilibrium, right? And the localized cysteine chemistry acts as a kinetic driver. It actively participates in setting the thermodynamic landscape, helping to physically push the receptor energetically from an inactive state to an active one. Let's move to the modeling side and see how this builds, because validating these complex dynamic shifts requires incredible precision. This is exactly where modern computational tools, like molecular dynamic simulations, come into play. By computationally modeling these receptors, researchers can bridge the gap between atomic-level cysteine chemistry, which is happening on the scale of angstroms and picoseconds, and the massive macroscopic pharmacological effects we observe in the lab and the clinic. It literally allows us to visualize how a single sulfur atom helps a massive receptor protein breathe and move in real time. Next up, the open research frontier. These are the pressing questions our field must answer next. As researchers standing right on the edge of this expanding field, the horizon is incredibly exciting. The integration of the redox code with GPCR biophysics has opened up entirely new avenues of inquiry. Here are the bleeding-edge questions we still need to figure out. First, tracking these signals in real time requires way better spatial and temporal resolution of hydrogen peroxide microdomains. Second is the competition factor. If a cysteine can be modified by multiple reactive species, what rules dictate which modification wins in a live cell? Third, we really need to nail down the true fractional occupancy of persulfidation in resting versus stimulated cells. Fourth, linking back to GPCR biophysics, the exact mechanics requires strict characterization. Is it covalent chemistry, hydrogen bonding, or both that's modulating these receptors? And finally, the ultimate challenge, bridging these massive datasets from chemoproteomics directly to functional biology and native cell membranes. The reality is, what we currently know about cysteine is really just the tip of the iceberg. Whether it's developing brand new covalent drugs that target these precise sulfur switches, or mapping entirely new signal transduction pathways, the opportunities to fundamentally change our understanding of cellular biology are absolutely massive. So as we close our researcher's notebook for today's explainer, I want to leave you with this provocative thought. The reactive cystinome is vast, dynamic, and full of secrets. Which code will you crack next?